And I hope that you really enjoy this course and that you get a lot out of it. So now I'm going to start. So sorry, I'm going back and forth here to have a look at where we are. I'm here. So now I'm going to share my screen. And here. Oh. Oh, Gabby, you mind moving Nina around? Thank you. Hi. There we go. Okay, excellent. Today we're going to talk about working with light and with fluorophores. So first, I want to hit the nail on the head before I even plant the nail. So I'm going to tell you some concepts here that are very general and are sometimes difficult to understand or difficult to grasp. And so I hope that throughout the course it will make sense. So the first thing is that we start by image analysis. And so segmentation is one of the keys of image analysis. With AI especially, it's not the only key, there are other keys, but it is a very important step in image analysis. So we need to remember that the goal of everything we do before we go to the microscope, so conceptualizing an experiment, growing animals and cells and collecting patient samples, and then perfusing and cutting and fixing and staining, acquiring the images, and designing and running the image analysis is to extract numbers. This is what we want, and this is because we are scientists, and this is the way we can compare things. So we extract numbers from the images for scientific comparison. So the most challenging step in many image analysis pipeline is the segmentation, very often. And so what is segmentation? Segmentation means that you're separating individuals' areas or objects from each other. So when you segment an image, you're creating a mask on top of the image, and the mask only shows you the areas that you're interested in. So here is an example. We have this original image that shows some cells with vesicles, and there is some sort of a background as well. So we are interested in the vesicles, but we're not interested in some sort of background, a general background that is also in the cell. What are we going to do? We're going to create a segmentation mask. And this is what the mask looks like. You can say that it looks a little bit like a completely black mask in which you have made holes at the places where the interesting bits are, right? And then when you overlay the original image with the segmentation mask, all you see is the interesting areas, a little bit as if you were seeing through these holes. And so this process is called segmentation, and it's a very important step in image analysis. So throughout the course, I'm going to talk about, this makes the segmentation harder, this helps with the segmentation. This is what I mean. I mean, it makes it easier or harder to make this mask. So here are a few key concepts that are a little bit challenging in microscopy. They are microscopy artifacts that are not often not enough talked about, say. So refraction is one of them, and then bleed through, saturation and underexposure, and undersampling. So in this course, we're going to talk about all these, and these are artifacts, so we want to avoid them. Another concept that is a little bit challenging is that the final resolution in the image depends on several things. It depends on the optical resolution, which is the resolution that the objective and the light wavelength delivers. The digital resolution, which is the image pixel size, and then the sample quality that leads to the contrast. Okay, so we're going to talk about these different types of resolutions and as well as the sample quality and the contrast in this course. And one important aspect of designing a microscopy experiment is to start with the end. So we start with the image analysis and we move backwards. This is called reverse thinking. And you can see here this reference that I have, and it's also in Canvas if you want to look at it. it's a very interesting article that crystallizes the way you want to organize your experiment. You first you think about what you want to measure. These are the metrics of your experiment. What you want to measure, and then you decide how you're going to analyze, and then you design your sample. You choose the microscope, the objective, the imaging settings, and you run the experiment, and then you learn at each step, and then you go again and do one more iteration. And this is the way you run your experiment, not the other way around. I'm going to prepare all my samples and then I'm going to image everything and then I will think about the image analysis. This does not work. And quite interesting concepts, which I will explain today. Hopefully you will understand what we mean. The image of an object is never and cannot be like the object. What we image is the interaction of light with the sample and the microscope. So the lectures that we're going to talk about now will explain some behavior and properties of light. But remember that what we're imaging is not the object, it is the light that comes from the object, and therefore the image can never be the same as the object. So now I'm going to talk a little bit about nature of light, so that to put a few stones in place. So light is an electromagnetic wave. What does that mean? Oh, a charge, so a small charge that does not move, produces around itself an electric field. But if the charge starts moving with a constant speed, then it produces an electric field and a magnetic field. And now, if the charge accelerates and decelerates, so it moves, but with varying speeds, it goes faster and slower, faster, slower, then it produces an electromagnetic wave which starts propagating through space. And this means that the electric field and the magnetic fields become coupled. Okay, so light is an electromagnetic wave because we have some point charges that are actually moving through space and that accelerate. They are moving in a way that they accelerate and decelerate all the time. So they create an electromagnetic wave that propagates through space. And this is why we can see light from far away from the source. So in this little diagram, you can see the electric field and the magnetic field. They are coupled, so they oscillate in unison. And then here we have the travel of the light. So the light is then moving through space. Because of this, there are several ways to represent light. And in this course, we will jump from one to the next. So here is, for example, one way to represent light. Light is the wavelength of light. So this is a number, and it corresponds to the distance in nanometers in general between the crest of. So I'm saying that it oscillates, right? So between the crest of one oscillation to the next. So this is the wavelength. We talk a lot about the wavelength in general. The wavelength is abbreviated as lambda. So when you see this, it means wavelength of light. Another way to represent light is ray optics. So in ray optics, all we represent is the direction of light. We don't say anything about the wavelengths. You don't say anything about the fact that it oscillates or anything. We just represent the direction. This is ray optics. But another way, again, to represent light is with the wave front. So in the wave front, we will draw a line like this on each of the top of the waves. And you can see here we have a flat wave front, so the lines are parallel and they're straight. But in here we have a curved wave front. Okay, so we're going to talk about these different ways. I will use each of them, wavelengths, rays, or wavefront in these lectures. And there's also energy associated with light. So light is an electromagnetic wave, which is associated with energy. But the energy is not evenly distributed everywhere. So there's like a certain probability that the energy is going to be more in some places of others. And these places are actually called photons. So at some places where the energy is concentrated, then it's a photon. And you can see in here a very broad range of electromagnetic waves. And I was just wondering, Gabby, if you are recording, just in case.
Yep.
Thank you, thank you, thank you. So yeah, we have a very broad range, and you can see in here the cosmic rays and the gamma rays and the X-rays, they are very much in the short wavelengths. We have very, very tiny wavelengths in here. And then if we continue, we get UV, infrared. Here we have the visible spectrum that we will talk about. And then if we continue into longer and longer wavelengths, we end up in radio waves. And you know that radio waves, they go through us. So they're very, very long waves, and they go through buildings. So you can get the radio inside the building as well. So the building doesn't block the radio. So that has a relevance also for us, and I will talk about this right now. But if we look at the visible range in here, we can see different colors, and we start in the UV, and we go from more or less 400 nanometers to 700 nanometers. But we have different, for the different electromagnetic waves, we have different properties. So if we go to the short wavelengths, the light, the electromagnetic wave is more energetic. It's also more damaging, and you don't know this because the UV is very damaging for us, right? But we have a higher resolving power because of the wave being so the wavelength being so short. But it's blocked very easily by protections. For example, if you wear a t-shirt, you're going to be protected from the UVs from the sun, right? So it's absorbed immediately. So it's less penetrant. And now if we go to the other side of the spectrum, we have less energy in the light. So it's less damaging, but it has a lower resolving power because the resolving power is limited by the wavelength. So we have very long wavelengths, so we have a lower resolving power. I will talk about this now. However, these wavelengths are very penetrant, and so this is why radio waves can go through buildings. So the wavelength of the light determines how energetic, penetrant, and damaging the light is. So that works also for our precious samples, and also how we perceive its colors for our human eyes, but also for filters, which we will talk about today a lot. And the maximum resolution in the microscopy image. So now when we talk about resolution, let's have a definition so we are all on board with that. What is the definition of resolution? It is the shortest resolvable distance between two objects. So the shortest distance that we can have between two objects, and we can still say it's two objects, and they don't like fuse and we think it's one object, right? So the resolution is a distance. It's in nanometers, it's a distance. Never say, I have 500 pixels, this is my resolution. This is not correct. Unfortunately, this has been sort of the case with like TV and so in TV commercials, they say number of pixels. This is not a resolution, right? This is a number of pixels, but the resolution is actually a distance in nanometers. And that also means that because we want to be able to separate two objects when they're very close to each other, we can still separate them. We have a high resolution. So a high resolution means a short distance. Okay, so when we talk about the resolution, a 225 nanometer resolution is a better resolution, is a higher resolution than 450. So a high number in the resolution means a bad resolution, right? We want a small number of the resolution. And then there is something that you may have heard before, isotropic resolution. It means the same resolution in X, Y, and Z. It's called isotropic resolution, so the same in all dimensions. So this was a little bit of definition. So now in microscopy, we're mostly imaging in the visible range. We step a little bit in the UV for excitation, for example, and then nowadays we image more and more in the near infrared, so more towards the 800. So we've extended, but this is pretty much where we are staying in light microscopy. Okay, so oops, here we go. So the visible light is more or less is between 400 and 700, and we can see what changes between these is the wavelengths. So we have very short wavelengths towards the blue and the violet, and as we go towards green, yellow, and red, the wavelengths get longer. Here, 700 nanometers. So white light is a mix of all this in different proportions. But if you have several colors, our eyes will experience it as white. It's basically a mix of different wavelengths is white light. The different light sources that we use in microscopy emit different ranges of wavelengths. So the different light sources are not equivalent at all. They're actually very different from one to another. For example, a mercury lamp is going to emit light throughout the whole visible spectrum and even like quite far in the UV. But a few wavelengths are much, much, much brighter than others. So this is the type of profile that you get with a mercury lamp. If you use lasers, lasers are very monochromatic. So they emit, like if we say a 488 nanometer laser, we will have only 2 or 3 nanometers before and after 488. So it's a very monochromatic. LEDs have a quite wide, like sort of narrow and wide, if you want to say, range of wavelengths. So they're going to emit sometimes 30, sometimes up to 60 nanometers. So for LEDs, we will need to narrow this down to make it more specific than 30 or 50. But it's definitely narrower than a mercury lamp. So when you use a mercury lamp, you need only one mercury lamp. But when you use LEDs, you need several of them to cover the whole wave. Whole range. Oops, sorry, I go too fast here. Now, if we look at the sample side, scattering is a word that we use quite often. Oh, my sample is scattering. So in homogeneous matter, light travels without being disturbed. It will just go straight and it will not slow down. So it keeps on going. But in heterogeneous matter, like for example, our samples, there are particles inside the, lots of particles inside the sample. You have like some proteins and then you have DNAs and lipids and everything. And all the particles that are larger than the wavelength of light, so lambda, scatter the light. So it splits it and disperses it in all directions. You can imagine this is not very good for imaging. So scattering is actually a combination of reflection, refraction, and diffraction. We will talk about all this and we'll explain what it means. And that means that a lot of the light energy is spread. And so we get a lower intensity of excitation and also lower intensity of emission, and we get some haze. So this is the consequence of scattering. We don't like scattering. We would like non-scattering samples. So this is an image of the beautiful Sarek National Park in the north of Sweden. And what you can see in here, we have these shrubs on the lower side of the hills. And we are very likely to have the same shrubs here, but we do not see them. And the reason why we do not see them is because there is more air and more particles of water and dust and everything from the person who takes the photographs to this mountain and in here. So because you have more particles, more droplets of water and everything, light is scattered and we cannot see any details, and we have a lot less color, a lot less contrast in this area, and we do not see the shrubs that we see here very well. So this is exactly what happens in our samples. We have more scattering, less contrast, and less colors if the light is scattered, and because we have a non-transparent sample. So in fluorescence microscopy, unfortunately, both the excitation light and the emission light are scattered. So we get the double punishment, let's say, when the light goes towards the sample and when the light comes back from the sample, they are scattered in both directions. And the short wavelengths encounter more particles that are larger than them. So they scatter more. This is the reason why shorter wavelengths are less penetrant, because they scatter immediately as soon as they find something. And so this is the reason why the long wavelengths are less disturbed and they are more penetrant. So if you have a thick sample that is not so well cleared, for example, a long wavelength will go deeper into the sample than a short wavelength. So these type of things are things that you need to think about when you prepare a sample, when you decide which fluorophore you're going to use. Another thing to consider is absorption. So some samples absorb light very often, not always, but they do, some very often. And different materials absorb different wavelengths in a different way. So the wavelengths that are absorbed, so the sample will absorb some of the light, and the rest is going to escape the sample, whatever is not absorbed. And this is what determines the color of the sample. The light does not actually is not absorbed. So, for example, in here in the Swedish archipelago, we have the sunlight, and what we can see in here is the reflected sunlight. So the reflected sunlight has the same color as the sun. It's yellow, or white yellow, and that is because it has not gone through the water. It is bouncing on the water, and so what we're seeing is the same light that comes from here. It also is just reflected on and it has the same color. But in here, what we can see is the light that has gone through the water, and the water is absorbing some of that light, and it rejecting other type of light, and it's blue. So the light that comes from the water is going to be, so all the red and all the greens are going to be absorbed, and only the blue escapes, let's say, from the water, and this is why the water is blue. So whatever is not absorbed is going to show the color of the objects. Right? So it's the same thing exactly that happens in our samples. And now if I take a torchlight and I put it behind my hand like this, so this is white light. So I get white light, I bring in a lot of wavelengths, I put it behind my hand and I take a picture. What I see here, even though I'm shining white light on the other side, what I see here is red light. And why? This is because my blood absorbs all the blue and green light. And so what is left is red, and this is what comes out on the other side is red, and this is why I see that as red. Okay? So in tissue samples, the shorter wavelengths are absorbed faster, so blue. They scatter more, and because they're more energetic, they warm up the sample more. So we have more problem with shorter wavelengths than with longer wavelengths when we image tissues. So if you use live samples, you would like to not image anything that is blue. So this is a simple consequence of knowing a little bit how light works, but it's actually very important. If you have live samples, use long wavelengths, not short ones. Oops. This was a very simple introduction to light and how it works, and there are consequences for us, for light microscopists. So light is an electromagnetic wave. It's represented by its wavelength, its rays, or the wavefront. And the visible range is between 400 to 700 nanometers. And the short wavelengths are less penetrant in biological tissues. They get blocked very quickly. They are more damaging, very energetic, but they have a higher resolving power, whereas the longer wavelengths have the opposite. Scattering is a problem in microscopy for both the excitation and the emission light. And we lose the intensity. As the light is scattered, we lose intensity and we lose the contrast in the images, which all these things that we do not want. So we want to try and make a sample that is not scattering, and we will talk about this later in the course. Okay, so this was the first little lecture, and I'm going to look at if I'm very late or not. No, I think I'm okay. I'm actually early, so it's good. So now I would like us to do one of the quizzes. So for the students, you can go to Canvas and have a look at the quiz called Nature of Light. And throughout the quiz, so I can do it here actually, and then throughout and then in the future we will go to the other Zoom, but this one is very simple, so I just wanted to show it like this. Here it is. Right. So there are two questions. We're going to do this one live, and then in the future, we just go to the workshop Zoom whenever we have quizzes together. But this one is just the first one. So I need to count the nuclei in a culture of live cells, and I know that my sample is sensitive to light. In my lab, there are two nuclear stains. One is called Hoechst, which pretty much everyone knows, and DRAC5 is another one. Many people do not know that, but they do the same thing. They label the nuclei. Which one should I choose? So in here, you can pick if you want to label pick Hoechst, or you want to use DRAC5. And these are the excitation spectra for DRAC5. The pink one is DRAC5, you can see it here. And the blue one is Hoechst. So you can see here the wavelength. And here you can see the filters that we are going to use. So please let me know, would you choose Hoechst or DRAC5? You can pick it in the drop-down menu, but you can answer as well, so that too.
Yeah, I think that if it's live cells, as you mentioned, it's better red.
Better with the red. Would anyone have picked blue? So the long wavelengths are more penetrant, and they are less damaging. So we definitely want to get the red. Okay, right. So then the next question, Blick, if I can get to my question here. Sorry. Here it is. The next question is, I need to count RNAs in a fixed sample, and the dots are very crowded, so I need to image with the highest possible resolution. Which fluorophore shall I attach to the RNA probe? Shall I use DS-red or brilliant violet for 4:1? So this one is the brilliant violet, and this one is DS-red. Which one would you choose to get the highest possible resolution?
The violet.
The violet. Okay, is everybody okay with that? Simple quiz. But yes, exactly, it is the violet. And so when you submit the quiz, you get the answers that I would have given. So throughout the course, we're going to do lots of quizzes like this to try to help crystallize everything you learn. I'm going to continue to, so I'm just looking at my, okay. We have, I'm just a tiny bit ahead of time. We're going to take a tiny break, and we will start again at 45, so in 6 minutes. And if you guys have any questions, you're welcome to ask.
Thank you.
Welcome. Let me see. It's a bit difficult to navigate.